The overall goal of this method is to rapidly and reliably guide muscle fiber quantification in whole rat muscles in an automatic and user-independent manner using an open-source platform.
This method can help answer key questions in the neuromuscular field about the effects of aging, disease, or trauma on skeletal muscles. The main advantage of this technique is that it can be used to reliably analyze muscle fiber populations and to generate high-quality images of muscle cross-sections.
After sufficient thawing, carefully rinse the specimens in PBS supplemented with Triton X, or PBST, for one 10 and one 5-minute wash. After the second wash, air dry the sections for 2 minutes. Then, use a hydrophobic pen to encircle the individual cross-sections. After another 15 minutes of air drying, block any nonspecific binding with PBST supplemented with goat serum for 1 hour at room temperature. Next, wash the slides and add the appropriate primary antibody cocktail to each sample for a 1-hour staining at room temperature protected from light. At the end of the incubation, rinse the slides with one 10 and one 5-minute PBST wash. Then, label the muscle sections with the appropriate secondary antibody cocktail for 1 hour protected from light. After washing the slides 2 times in PBST, as demonstrated, dry the slides off and then stain the nuclei with a DAPI nuclear staining kit according to the manufacturer's instructions. Remove the excess DAPI with a 1-minute PBST.